So let's begin with the packaging of DNA into chromosome. How this DNA is packaged into the chromosome. So DNA during interphase, when the cells are not dividing, DNA molecule exists as nucleoprotein complex called as chromatin when they are not in the division state. And these chromosomes, they are much shorter than the DNA molecules that they contain, and the average human chromosome is around 5 centimeter of DNA. Okay, so there has to be organized packaging system which is required to fit the DNA molecule into the chromosome, and each chromosome consists of a single enormously linear long DNA molecule associated with proteins. So chromosome consists of what? DNA molecule plus proteins which are folded and packed and into compact structure. What kind of proteins? So these proteins, they are divided here into two classes, histone and non-histone chromosomal proteins. So total mass of histones in chromatin is about equal to that of DNA. They are equal, mass are equal, and histones, they are responsible for the basic level of chromosome, most basic level of chromosome organization. And the nucleosome, which is a structural unit of chromatin described by R. Kornberg in 1974. Okay, so nucleosome provides the first level of DNA packaging. So when DNA attaches with the histone protein, it forms nucleosome, which is first level of DNA packaging, and gives beads on a string, like thread with have a beads. Okay, so this is kind of histone, and this organization, the first level of organization, is known as beads on a string structure. So nucleosome contains around 200 base pair of DNA associated with histone octamer, and which consist of two copies each of H2A, 2B, 3 and 4. Okay, H3, 4, like kind of they form a tetramer, two copies of each, flanked by two H2A and H2B dimers. Okay, so these are known as core histones. So these core histones are rich in two amino acid, basic amino acid arginine and lysine, and the positive charge on these two amino acid, this neutralize the negative charge on DNA backbone. So these 8 proteins form a barrel shaped core octamer with the DNA around wound around the outside, and which is forming 1.65 turns in a left handed coil. So this histone, like which is coiling the DNA around itself, it is wrapping the DNA 1.6 turns in a left handed coil. And treatment with micrococcal nuclease initially cleaves the DNA between the nucleosome. Okay, it will cleave the DNA between the nucleosome, and mononucleosomes which have 200 base pair of DNA. On further digestion, the length has been reduced to 165 base pair, and this is reduced to the length of the DNA of the core particle 146 base pair. So when they are treated with micrococcal nuclease, the DNA will be cleaved between the nucleosomes. Okay, and nucleosomes which have 200 base pair DNA, when it is digested, the length of the DNA is reduced to 160 base pair, and finally reduced to the length of the DNA of core particle, which is 146. So 200 to 165 and 165 to 146 base pair. So you can see in this picture mononucleosome, which is wrapping around the DNA. When it is trimmed with micrococcal nucleus, uh, it is trimmed, and the length is initially 165 base pair, and then it generates the core particle with 146 base pair. So each nucleosome is separated by 50 to 70 base pair of linker DNA. So if this is one nucleosome, this is another nucleosome, this is another nucleosome, they are separated by linker DNA, giving the result giving the repeat length of 190 to 220 base pair. So changes in the length of linker DNA account for the variation in the total length of nucleosome DNA. So when the length of the linker DNA is changed, there is a variation in total length of nucleosomal DNA. H1 is associated with linker DNA, okay, and may lie at the point where DNA enters. So this lies at the point where DNA enters and leaves the nucleosome. So this all is asked in the exam, that histone is a octamer, each have two subunits, and H1 is attached with a linker at the point of the start, and this is the structure of a nucleosome where a DNA is wrapped around nucleosome. So then there is a group of additional histones. They are closely related to one another and collectively called linker histones. Okay. So then there is linker histones, another group of histones. Okay. And in vertebrate, these includes H1AE, H1 kind of 0, H1T, H5. So the single linker histone, it is, one is core histone, one is linker histone. Single linker histone is attached to each nucleosome to form chromatosome. So linker histone plus nucleosome is chromatosome. Okay, but the positioning of this linker histone is not known. So among histones, H3 and H4 are the most conserved proteins known, very important. H3 and H4 are the most conserved known, okay. H3 and H4 are the most conserved proteins known, and H1 show appreciable variation between these species. There is a variation in H1, but H3 and H4 are kind of conserved, and in each fish sperm and other sperm, protamine is present instead of histone. Protamine is a polypeptide consisting mostly of amino acid arginine. So instead of histone, protamine is present in case of fish sperm. Okay, and some other sperms. So what are the properties of histone proteins? They are lysine or arginine rich. H1 is what? H1 is lysine rich. H2A, H1 you can see it's the highest molecular weight is H1, which is associated with DNA, linker DNA. H2A is lysine, slightly lysine rich, 14,000. H2B is also slightly lysine rich, 13,800 molecular weight Dalton. H3 is 15,300 arginine rich. H4 is also arginine rich. So this first level of organization was nucleosome formation. The second level of organization is the coiling of the series of nucleosome into helical array. First was, it was beads on a string structure. This was first. Second is this one. In this case, there is a helical array to constitute the fiber of 30 nanometer. Okay, 30 nanometer fiber is formed, and it has 6 nucleosomes for every turn. Okay, for every turn 6 nucleosomes are there. For every turn 6 nucleosomes are there. Six nucleosomes are there, and the exact way in which nucleosome associate to form 30 nanometer fiber, it is not known. But how many chromosomes are there? How many nucleosomes are there? 6 nucleosomes are there. Okay, and the exact way which 30 nanometer fiber is forming the nucleosome is not known, but there are several models proposed. The most popular model is the solenoid model. So first was beads on string, second is a solenoid structure, 6 nucleosome in a turn, condensing to form chromatin fiber. So first was chromatin formation. Number 1 was chromatin formation. Number 2 is chromatin fiber formation. Okay, where each nucleosome associates with H1 molecule. Okay, and the fiber coils into solenoid structure with the 2 is solenoid structure. So you can see in this picture, very important for any biology exam and CSIR of course. So first is the double helix, 2 nanometer, then beads on a string structure, first level of chromatin organization, 11 nanometer, then solenoid structure, 30 nanometer, where 6 nucleosomes are forming a kind of ring, and after solenoid structure 30 nanometer there is 300 nanometer. Okay, there is 300 nanometer which is forming what, that's a kind of forming loop. So 300 nanometer, then it is 700 nanometer where is a more condensed form is there, then lastly 1400 nanometer. So in chromosome 1400 nanometer is there, where DNA is packaged into metaphase, that is 10,000 fold shorter than its extended length, 10,000 fold shorter. Okay, so first stage was what, first stage was DNA, then it was like 11 nanometer, first is 2 nanometer, then 11 nanometer, then 30 nanometer, 300 nanometer, 700 and 1400. Okay, so this was very interesting, the chromosome packaging was very interesting. Coming to histone modification, so all the histone proteins, they are kind of chemically modified, and there is some structural changes which occurs at the time of replication and transcription. The most common type of chemical modification are given here, acetylation and methylation. Okay, so each of the histone protein which makes up the nucleosome core, it contains a flexible amino terminus at 11 to 37 residues extending from the fixed structure of nucleosome. Okay, so histone protein which makes up the nucleosome core, it contains an amino terminus, okay, having 11 to 37 residues extending from the fixed structure of the nucleosome. The N termini are called histone tails. Okay, so N termini having 11 to 37 residues extending from nucleosome have histone tails, called histone tails, and to this histone tails acetyl groups are added to the lysine amino acid. Okay, this histone tails also have what, histone tails also have lysine residues, and acetyl groups are added to the lysine amino acid in the histone tail. And acetylation, what acetylation is doing, acetylation is reducing the affinity of the histones for DNA. Acetylation is reducing the affinity of the histones to DNA, so that the interaction between the nucleosome leads to the formation of 30 nanometer fiber. So what acetylation is doing, it is reducing the affinity of the histone for DNA. Okay, so that there will be no interaction between nucleosome. Okay, so this histones in heterochromatin, they are generally unacetylated, where those in euchromatin are acetylated. So what it is doing, this acetylation is loosening up the chromosome, so that they can be expressed, more proteins and transcription factors can come and bind to that region, so that those region can be expressed. Unacetylated region are silenced, inactive. Acetylated region are active. So euchromatin are acetylated, hetero are unacetylated. Next is phosphorylation. So phosphorylation, kind of not much known about phosphorylation, but it has significant impact. Phosphorylation of histone H3 and H1 has been associated with the formation of metaphase chromosome. So phosphorylation is seen in H1 and H3. Okay, H1 and H3. Methylation is also in arginine and lysine at the N termination. So methylation is associated with inactivity, acetylation with activity, methylation with inactivity, and the sites that are methylated includes 2 lysine of H3 and an arginine of H4. Okay, so methylation sites are 2 lysines of H3 and 1 arginine of H4, inactive chromatin is methylated on 9 lysine of histone H3. So whenever 9th lysine is methylated on histone H3, it is in activation. Okay. So next, coming to metaphase chromosomes. So that was kind of different kinds of modification which is seen in histones. Phosphorylation is rare in H3 and H1. Next, coming to metaphase chromosome. So once 30 nanometer fiber is formed, which is a major type of chromatin in the nucleus during interphase. Okay. And when the nucleus divides, what will happen? There is a more compact form of packaging which will be formed, and highly condensed is seen in metaphase chromosome. Okay, which is 1 to 1 by 10,000 of its stretched out length. And when the loops are formed, containing around 20 to 100 kb of DNA per loop, are attached to central scaffold. So after this 30 nanometer region, there is a formation of scaffold, and this consists of non-histone acidic proteins, notably topoisomerase 2. Okay, so loops of 30 nanometer fiber containing 20 to 100 kb of DNA per loop, they are attached to a central scaffold. And this consists of non-histone acidic proteins, notably topoisomerase 2, an enzyme which has interesting ability to pass one DNA double helix through another by cutting and sealing both the strands of DNA. So once the DNA in the loop structure, once the DNA, which each loop consisting of 20 to 100 kb of DNA, when they are attached to the central scaffold, in that kind of structure, which consists of non-histone acidic proteins, namely topoisomerase 2, which is an enzyme, it has what will happen in loop structure. Okay, in that case there is the nick. Okay, kind of you can say, where one strand will pass to pass DNA double helix through another. Strand can pass through another strand by cutting and sealing, and this topoisomerase 2 and some other proteins are known to bind to AT rich sequences, and the chromatin loops may get attached by several hundred bases of highly AT rich DNA. Okay, scaffold attachment regions SAR. So beside histone and topoisomerase, there are structural maintenance of chromosome proteins. They maintain the condensed chromosome structure. So what we studied that once loop is formed, it will bind to what? It will bind to the scaffold, and in this scenario, there will be one non-histone protein named topoisomerase, which will help one DNA double helix to pass through another, leading to more condensed state, and some other chromatin proteins which bind to AT rich sequences, and the chromatin loops may attached by stretches of several hundred bases of highly AT rich DNA. So what will happen? There will be some other chromatin proteins which will come into the role, and they will attach themselves to AT rich. Okay, chromatin proteins will attach themselves to AT, so that more of AT rich kind of loop will come in contact with each other, and as a result of which several hundred base pairs of highly AT rich will become attached. Okay, and the chromatin loops may be attached by stretches of several hundred bases of highly AT rich DNA. And another proteins, structural maintenance of chromosome, SMC proteins, they will maintain this condensed structure. So how we understood? First is 30 nanometer, then attaches to the scaffold, then what will happen? Topoisomerase will come into play, which will bind. Topoisomerase will help one DNA helix to pass into another, more condensation will occur, and some other chromatin protein which will bind to AT rich sequences, as a result of which the chromatin loops may attached by several hundreds pairs of AT rich. So AT rich will be attached, and kind of several loops will be attached to AT, as a result of which there will be more condensation, and then there will be condensation proteins, maintenance of condensation chromosomes. Some proteins will be there which will maintain the condensed state, and eukaryotes have two SMC, which is structural maintenance of chromosome proteins. It has two, cohesin and condensin. What is the role of cohesin? Which play an important role in linking sister chromatids immediately after the replication and keeping them together, and condensins make the chromosome highly condensed. So linking two sister chromatid, cohesin, after just immediately after replication, and condensin is condensing the chromosome. So in the chromatids of metaphase chromosome, the loop scaffold complex is compacted yet further by coiling. So the loop scaffold complex will further be compacted by coiling. So you can see the typical structure of a metaphase chromosome. Each chromosome has replicated and comprised of two chromatids, and each chromatid, each containing one of the two identical DNA molecules. It has its own DNA, both are identical, and this is a centromeric region. This region is a centromeric region which will attach the two chromatids, and which is required for the separation in late mitosis. And there is a telomeric sequence at the end. Which prevents chromosome shortening. So next we come to polytene chromosomes. They are discovered by Balbiani in 1881 in larval salivary glands, known as giant chromosomes at that time, and they are specialized interphase chromosomes present in certain insect cells and differ with other mitotically dividing cells and those undergoing endomitosis. So in what ways do they differ? So this polytene chromosome, they are associated with the elimination of entire mechanism of mitosis after each DNA doubling. So there will be no mitosis, okay, after each doubling, as a result of which cell cycle consists of just two periods, synthetic and intersynthetic. Okay, there will be no mechanism of mitosis won't be there. Okay, and there will be just two periods, synthetic and intersynthetic. Second, at the end of each replication period, daughter chromatids do not segregate, rather they remain paired to different degrees. Third, polytene chromosome formed are incapable of being involved in mitosis. Okay, they do not show mitosis. Daughter chromatids do not separate. Fourth is the nuclear membrane and nucleolus remain intact during consecutive DNA replication cycles. No dissolution of nuclear membrane. So polytene chromosomes, polyteny, it has been studied in the salivary glands of Drosophila larvae, in which the DNA of each 4 Drosophila chromosome has been replicated through 10 cell cycles without any separation of daughter chromosome, so that 1024, which is 2 to the power 10 identical strands of chromatin are lined side by side. So when they are viewed under light microscope after staining, it was found that distinct alternative dark bands and light bands, interbands, are visible. Okay, and 95% of the DNA is in bands and 5% in interbands. And the chromatin in each band appears dark, either because more condensation is there than in interbands, or it may have higher proportion of proteins. Okay, maybe due to dark is maybe due to condensation or due to higher number of proteins, or both. But both bands and interbands, they contain genes. So bands are the sites of gene expression expand to give puffs, which is Balbiani rings. So those bands They are the site of gene expression, which shows puffs where the genes are expressed, and these puffs are known as Balviani rings. And this consists of a region in which chromosome fibers unwind from their usual state of packing in the band. Okay, so the puffs are the region where RNA is being synthesized because expression occurs here, and the characteristic pattern of the puff is found in each tissue at a given time. Okay, so puff is not throughout, it is where the gene needs to be expressed. So organ containing cells with polytene chromosome, they involved an intense secretory function which is accomplished during a short time against the background of rapid growth. Okay, so when there is rapid growth, you will find that those organs cells where polytene chromosomes are there are, as a rule, they are involved in intense secretory functions. Okay, and which is accomplished during a short time against background of rapid growth. The features of polyteny provide the conditions necessary to accomplish these functions. What functions? That is, during a short time intense secretory function is there, and during a short time against a background of rapid growth. Okay, they are involved. Why polytene chromosome is formed? Because those cells are involved in intense secretory function and they have to do it in a short period of time, so they have to grow rapidly. Okay, next, similarly to this, there is a lamb brush chromosome. Lamb brush chromosome was first observed by Fleming in 1882 in amphibian oocytes, and it occurs in the diplotin stage. Okay, that was, I think, in interphase. It is in interphase. Okay, but lamb brush is in diplotin stage. In the diplotin stage of the meiotic prophase during oogenesis in oocytes of many animal species, the lamb brush chromosomes, they are meiotically bivalent and consist of two pairs of sister chromatids held together by chiasmata. Okay, two pair of sister chromatids held together by chiasmata, where the loop is shown in the figure. Okay, so here it shows, this figure shows that there is a segment of DNA that is being actively transcribed. Okay, which is actively transcribed. The lateral loops which are formed, they extend in pairs from each sister chromatid, and these loops are surrounded by a matrix of ribonucleotides, ribonucleoproteins. Okay, they are like kind of surrounded by ribonucleoproteins. Okay, which contain nascent RNA chains. Okay, the loops are surrounded by ribonucleoproteins which are nascent RNA chains. So lamb brush chromosomes are thought to assist in fulfilling the high demand for transcripts during oogenesis. Okay, so most of the DNA in each chromosome remain highly condensed in the chromomere. Okay, and the two chromosomes shown consisting of two closely opposed sister chromatids, and this four strand structure is a characteristic of diplotin stage of meiosis. One is paternal chromosome, one is maternal. There is a loop which extends from both sides, and this is where the to fulfill the high demand of transcripts during oogenesis. So in the next video we will start with DNA replication.